So today we are going to talk about the basic concepts related with the epithelium. Epithelium or epithelial tissue or epithelia. Right? Now, human body consists of primarily 4 basic tissues. What are those 4 basic tissue? Yes, please. What are those 4 basic tissue which make our body? Yes. Of course, number 1 there is epithelia. Number 2 is connective tissue. Number 3 is muscles. Very good. And number 4 is neuronal tissue, right? Nervous tissue or neuronal tissue. So body basically consists of 4 tissues: epithelial tissue, connective tissue, muscles, which include cardiac muscles, smooth muscles and skeletal muscles, and neuronal tissue. Now, today's lecture is about the epithelium. So, who will define epithelium for me? Any idea? Epithelium is made of tightly packed, tightly opposed cells. Number 1 thing about epithelium is that it consists of tightly opposed cells which are very much, the membranes are very near to each other, and there is no vascular tissue in between. This epithelium is avascular. There is no vascular tissue in between. What is epithelium? Epithelium is collections of cells which are opposed together with very little material in between, right? And epithelial tissue rests on basement membrane which separates the epithelial tissue from other tissues. For example, underlying connective tissue. Is that right? So what is epithelium? Epithelium is basically made of cells which are very much opposed with each other, without much tissue in between, without much material in between, and without any vascular tissue in between, right? And now these epithelial cells are arranged basically in 2 fashion. That epithelial cells are arranged as long sheets of cells. Cells are arranged in sheets. So we can say membranous epithelium. What is that? Membranous epithelium. Or in some parts of the body, epithelial tissue makes glandular tissue. Epithelial tissue makes, yes, glandular glands, right? Glandular tissue or glands, right? In glands what happens, that epithelial cells, look, these are different epithelial cells. Again, they are very much attached with each other, and they in between this cell and the other cell, there is no vascular tissue, there is no other tissue. Is that right? They are again tightly packed cells, but they can act as glands and produce different kind of secretions. So we can say either epithelial cells are as sheets, right? They are as membranes or sheets, or they are folded. Epithelial cells are arranged in glandular pattern. Now, when epithelium is present as long membranes, these membranes are, number 1, covering the external surface of the body. Epithelial tissue or epithelia cover external surface of body. Of course, that is epidermis. Secondly, epithelial tissue also line all the internal closed cavities. Number 2, it also lines internal closed, yes, cavities. For example, it is lining the cardiac cavity, pericardial cavity, pleural cavity, and abdominal cavity is also called peritoneal cavity, right? Around the heart there is pericardial cavity, around the lungs there is pleural cavity, and in the abdomen there is peritoneal cavity. So these cavities are also lined by epithelial membranes or epithelial cells or epithelial tissue. And of course, your cardiovascular system, in which blood is circulating, cardiovascular system all arteries and veins and capillaries and cardiac chambers, all the cardiovascular system is also closed cavity. So cardiovascular system is also lined by epithelium. Very good. So cardiovascular system is also lined by epithelial tissue. Is that right? Then we come to third component is that epithelium also lines all the body tubes. Epithelium also lines the body tubes which communicate to the exterior. For example, tubes of respiratory system, tubes of gastrointestinal system, tubes of urogenital system. So it means all the body tubes which are communicating to the external, right, exterior, they are also lined by different type of epithelia. For example, yes, respiratory system, gastrointestinal system, and urogenital system. So let me repeat it. What is epithelium? Epithelium is special type of tissue in which cells are very much in proximity with each other. There is no much material in between the cells, right? And epithelial tissue rests on the basement membrane which separates the epithelial tissue from the underlying connective tissue. Epithelial tissue can be arranged as membranes or can be arranged as glands. Is that right? When it is arranged as membranes, right, then it is lining the body surface, external surface, as well as it is lining the internal cavities of the body, as well as it lines the tubes in the body which are communicating with the exterior. Is that right? So this is about the basic. Now, what are the functions of epithelial tissue? What are the basic functions of epithelial tissue? Yes, who will tell me? What are the basic functions? Of course, number one is protection. Skin, it protects you. So when we talk about function of epithelial tissue, number one there is, yes, protective function. For example, epithelium on the skin, which is epidermis, it protects you from abrasion, right, or injury. Second is secretions. Epithelial cell also produce secretions. For example, in the gastrointestinal system, let's suppose that here are epithelial cells. Now these epithelial cells, right, which are lining the gastrointestinal tube, these epithelial cells, they can produce different types of secretions. Then they also play a role in absorption, that when you are taking the food, when proteins are broken down into amino acid or carbohydrates are broken down into glucose, this is taken from lumen of the gastrointestinal system through the epithelial cells to the connective tissue and circulatory system, and going to the circulatory system. So what is its function? Number one, it can secrete different type of secretions. Number two, it can absorb also material. So its functions are protective, its functions are secretions, its functions are, yes, what is this? Absorption. Right? Epithelial cells can absorb the substances, they can secrete the substances, they act as protective layer on the external surface of the body or even within the body organs. Then, any more function of epithelial tissue? Yes. Any idea? There's one more function of epithelial tissue, very easy function. Look. Pardon? They talk to each other, they communicate to each other. That is not the function of, that is a feature of epithelial tissue. I'm talking about one special function that epithelial tissue can, yes, very good. That is sensory function. Some cells in epithelial tissue, some cells in some specialized epithelia can detect the stimuli. For example, there is olfactory epithelium in the nose and that can sense the smell, odor. Or there is epithelium related with the tongue which is making taste buds and that can work as taste detection system. In the same way, in the internal ear, there are special sensory epithelia in the internal ear which help you to, yes, hear, and they also help you to keep your balance. So what we can say that some specialized epithelial linings play an important role in, yes, detecting the different type of stimuli, right? Olfactory stimulus is sensed by olfactory epithelia. In the same way, for the taste stimulus is detected by gustatory epithelia on the tongue, and then there are sensory epithelia in the internal ear which can detect the, yes, movement to keep the balance, as well as they can detect the sound. Is that right? So basic functions are protection, secretion, absorption, and detection of sensation, right? Now we come to the different types of epitheliums. Epithelial tissue can be classified. I will go in detail of membranous epithelium now, right? I will go into detail of membranous epithelia. And when we talk about membranous epithelia, how are they classified? Membranous. Okay, rather membranous and glandular, we classify them together rather, right? So just we talk about, we are classifying epithelia. Now, to classify epithelia, basically it is on two basic criteria. Number one, what is the shape of cells? Shape of epithelial cells, right? Shape of cells, for example, some cells in epithelial lining, they are flat cells with a central nucleus. Is that right? Now this cell is a flat cell, and in this cell, if you look, yes, there is a central nucleus, but it is like a fried egg, right? Height of the cell is very little and width of the cell is very wide. Is that right? These type of cells are called, yes, these type of cells are called squamous cells. So those epithelia which are having flat cells, we call them squamous epithelium. Then there are other epithelial cells, and those epithelial cells have equal, yes, equal height and width and depth, right? Now such cells which have these features, right, these cells are like a cube. Because these cells are like a cube, so such epithelial tissue which is having cells like cubes, they are called cuboidal epithelium. Cuboidal epithelium. Then there in some other epithelia, cells are very tall. Cells are very tall, and in these epithelia, nucleus is near the basal area. This is the apex of the cell, this is the base of the cell, right? And nucleus is pushed near the base. And when any epithelial tissue has cells which are tall, where height of the cell is too much, but width of the cell is less, right? We say these cells are like a column. And because these cells are like columns, we call them columnar cells, and such epithelia are called, yes please, which epithelia? Yes, columnar epithelium. So there is squamous epithelium when they are flat cells, there is cuboidal epithelium when there is cells with almost equal height and width, and then there are epithelia which we call it columnar epithelia, in which fundamentally cells height is too much and width is less, right? So this is columnar epithelia. Am I clear? Now, so this is one way to talk about epithelium according to the cell. And then another way to talk about epithelia is according to the how many layers of cells are there. According to the layers of cells. Layers of epithelial cells. Right? Now, when we talk about layers of epithelial cell, there may be single layer, or epithelial tissue may have multiple layers of cells. Multiple layers. Whenever there is single layer, such epithelium is called simple epithelium. And if there are multiple layers, we call this is having stratification. So when there are multiple layers, we call it stratified epithelium. Stratified epithelium. Now, simple epithelium, if it has only, yes, now you have to tell me, it has only one layer with flat cells. What is this kind of epithelium? Yes? Simple squamous epithelium. What is it? Simple squamous epithelium. But if there is single layer of cells, but they are cuboidal, what is this? Simple, yes, cuboidal epithelium. And if cells are columnar, but there is only one layer of cells, what do we call it? What kind of epithelium is this? Yes, simple columnar epithelium. Important point here is, in simple epithelium, all the cells which are participating in epithelial lining, all of them are attached with what? Basement membrane. All the cells are resting on the basement membrane, right? In simple epithelia, cells have single layer and all the cells are attached with the basement or resting on the basement membrane. Is that right? But when we talk about stratified epithelium, right? What really happens in stratified epithelium, that there are multiple layers, right? If there are multiple layers, then all cells are not resting on basement membrane. For example, there is a basement membrane here, right? And this is multi-layer epithelium, and let's suppose these are different cells over here, right? And cells may slightly vary in shape from layer to layer. Now, this is not simple epithelium, it is multi-layered epithelium in which only the basal cell are resting on the basement membrane and other upper layer cells are not resting on the basement membrane. So such epithelium is called stratified epithelium. Now, stratified epithelium is named according to the cells which are present at the top. Now, the cells which are present on the top layer, they are squamous cells. So this should be called what kind of epithelium? Stratified squamous epithelium. What is it? This is stratified, yes, squamous epithelium, right? Now we look at another type of epithelium, that here the cells are again multi-layered, right? Cells are multi-layered, or epithelial tissue is multi-layered, and again we have to look what is the features of cells at the top. Now in the top layer, if you look at these cells, are these squamous cells or cuboidal or columnar? Yes, these are cuboidal cells, and because all cells are not resting on the basement membrane and it is multi-layered, so it is stratified epithelium, and as the top layer is cuboidal, what we will call this type of epithelium? Yes, stratified, very good, stratified, yes, cuboidal epithelium, because you have to look at the top, cuboidal epithelium. Is that right? Then you may come across another type of epithelium. Again, it is multi-layered. So we can say that this is also stratified, but here the top cells are not squamous and top cells are not cuboidal, rather they are, yes, columnar. So how you will name this epithelium? Very good. This is called stratified columnar epithelium. This is stratified columnar epithelium. Then there's another type of epithelium which is specially found in urogenital system, and in urogenital system that epithelium, you know, for example, the epithelium which is lining the urinary bladder, right? It is 4-5 layers of cells, but it has some special feature that when urinary bladder is full of urine and urinary bladder stretch out, the layers become more or less. Layers become less. And when you pass the urine out and urinary bladder becomes smaller, then the same number of cells are accommodating into smaller area. So number of cells in a layer will be in a tissue, the layer of cells will become more. Is that right? So such type of epithelium in which when it is stretched, there are less layers, and when it is, this is when it is stretched, and when it is not stretched, right, this is making more layers, right? This type of epithelium, that when it is stretched, right, as urinary bladder is full of urine and it is stretched, it is less layer, and when urinary bladder is empty and whole epithelial cells layers become more as it gets smaller, such epithelium is called transitional epithelium. Transitional epithelium, right? Transitional epithelium is only found in urinary system, right? In the ureter and calyces which drain the kidney, minor calyces which are draining the kidney, and ureters and urinary bladder and urethra. Now, what we have seen, that there are simple epithelia and there are stratified epithelia. Now we go into a little more detail of every epithelium. Simple squamous epithelium, where it is found? Simple squamous epithelium is lining the cardiovascular system, right? For example, it is your blood vessel system, cardiovascular system. The epithelium which lines here, the simple squamous epithelium is called endothelium. Endothelium, right? In the same way, simple epithelium also lines the respiratory system. In the respiratory system, when air reaches to gas exchange area, there is simple epithelium lining here in the alveoli. This helps in gas exchange. That oxygen will go from the alveolar area, from air bags to the blood, and carbon dioxide will come from blood to the air bags, alveoli. So this is also simple squamous epithelium, and endothelial cells are also simple squamous epithelium. In the same way, in the kidneys also, in the kidney there are special tubes. There are tubes which are called nephrons. In one kidney you have 1.2 million nephrons. And in the nephrons, especially in this area, which are filtration unit, they are flat cells, and these are also called, yes, simple squamous epithelium, right? Simple squamous epithelium is for filtration purpose here, for gas exchanges here, and for metabolite exchanging there. For example, glucose or amino acid from the blood go to the tissues, and maybe metabolic waste come back to the blood, right? So therefore, usually exchange epithelium. Simple cuboidal epithelium. Simple cuboidal epithelium is normally present in which tissue? Yes, simple cuboidal epithelium is normally present, number one, in the nephron. In some areas of the nephron, this is nephron. Nephrons are special tubes in the kidney which make the urine, right? So nephrons have in the filtration unit simple squamous epithelium, but here they have cuboidal cells. So nephrons may have simple cuboidal cells. And then in the same way, in the glandular ducts, many glands, their ducts are lined by simple cuboidal epithelium. In females, surface of the ovaries is also lined by simple cuboidal epithelium. Then we come to simple columnar epithelium. Classically, this epithelium is present in gastrointestinal system, right? Simple columnar is present in, yes, here. Gastrointestinal lining, stomach, small intestine, large intestine, and simple columnar is also present. What is this tissue? gallbladder. So simple columnar is also present in the gallbladder. After discussing this simple epithelium, right, I want to talk about some very special type of epithelium. Look here, there's a basement membrane here, right? Some cells are tall, other cells are not as tall. Now, this is an epithelium in which every cell is just reaching up to the basement membrane, but because cells have different heights, right? Because cells are different height, some cells, cells which are top, nuclei have gone at upper level, and cells which are basal area, the nuclei are at lower level. So when you make a section under microscope, due to the presence of nuclei at different level, it looks as if this is stratified epithelium, but actually it is not stratified because every cell is resting on the, what is this? Basement membrane. Right? So apparently it looks stratified epithelium, but because, why it looks stratified epithelium? Because some cells are tall and some are not tall, and nuclei are present at variable height. Is that right? So it is not truly stratified. So what we call it? What we call this type of epithelium? Yes, which looks like stratified, but it is not stratified. We call it pseudo-stratified epithelium. What is this? Pseudo mean false. It is stratified but false stratified, not a true stratified. Pseudo-stratified epithelium, right? Pseudo-stratified epithelium is found in trachea, right, and respiratory conducting pathway. In respiratory system, at the end, in the exchange area, there is simple squamous, but in upper part, this is pseudo-stratified epithelium, which is having usually cilia also, and they have some cells which release mucus secreting cells, right? These are the special features of pseudo-stratified epithelium present in airways and respiratory system. Then pseudo-stratified epithelium is also present in the epididymis. Epididymis is in the males, right? Epididymis. Do you have any question up to this? Okay, we come back now to this stratified squamous epithelium. Listen, stratified squamous epithelium has two variants. This is post-basement membrane. This is stratified squamous, but some of the stratified squamous, even though top cells are flat, but the uppermost layer cells, these are flat, but uppermost layer cells do not have nuclei. These cells are very flat and they do not have nuclei. They are just full of keratin. They are full of keratin. So this type of stratified squamous epithelium, which has at the top cells without nuclei and full of keratin protein, this type of stratified squamous epithelium is called keratinized stratified squamous epithelium. What you will call it? Keratinized stratified squamous epithelium. Now classically this keratinized stratified squamous epithelium is present on your skin, right? This keratinization help you to resist the injury or abrasions or mechanical pressure on the skin, right? So there is stratified squamous epithelium which is keratinized. We also call it stratified squamous epithelium which is keratinized and dry. Usually skin is dry until you don't make it wet, right? And we also call it stratified squamous epithelium which is cornified. So cornified or keratinized, it is one and the same word, right? Cornified, cornified. But look, if you compare these two epithelia, both of them are stratified squamous epithelia, but this is cornified or this is keratinized, but that is not keratinized because here the top cells having nuclei, and they are not having the flat cells without nuclei with keratin. So top cells are, even though flat, but they are having nuclei. So we call it what? Stratified squamous epithelium non-keratinized, or stratified squamous epithelium non-cornified. This is keratinized, that is non-keratinized. This type of epithelium is found where in the body? This is found in your oral cavity, and this is found in the vagina, stratified squamous epithelium which is non-keratinized, right? And then an esophagus, right? So oral cavity, esophagus, and vagina has stratified squamous epithelium non-cornified, skin has stratified squamous epithelium cornified, and then we come to stratified cuboidal epithelium. This is present in ducts of some glands, some glands that is having stratified cuboidal epithelium. And stratified columnar epithelium is also present in large ducts of the gland, right? And in the end, what is there? Transitional epithelium, right, which is multi-layered epithelium, but when it is stretched, layers become relatively less. When it is not stretched, layers of the cells become relatively more. This kind of epithelium is urine proof. It does not allow the urine to escape from urinary system and to reabsorb in the body. And this is called transitional epithelium, which is lining the calyces, ureter, and urinary bladder with urethra. Any question here? There is no question. Now we come to the real business, that is about the specialization of epithelial cells. Look, as we look at these epithelial cells, okay, I'll just look. Now, as you know, one side of the cells, epithelial cells, one side of the cell is resting on the basement membrane. Other side of the cell is, yes, free side of the cells, right? So this side of the cell, this is called basal domain of the epithelial cells, right? And this side of the cell, which is at the top of the cell, this is called apical side of the cell, or apical surface of the cell, or apical domain of the cell, right? This is apical domain of the cell, this is basal domain of the cell, and this side of the cell, epithelial cell, which is adjacent to the next epithelial cell, this is called, yes, lateral domain of epithelial cell, or we call it lateral surface of epithelial cell. So classically, epithelial cells have apical domain, which is the free side, and they are having lateral domains where epithelial cells are in contact with each other, and then basal domain where epithelial cells here are in touch with the basement membrane. Any question up to this? Now we will talk about that different areas of these epithelial cells, different domains are structurally and functionally and even biochemically specialized. For example, apical domain may have different functions and basal domain may have different functions, right? So I will talk now about the epithelial cells, different domains with specialized features, right? Because different surfaces of the epithelial cell have different type of features, so we call that epithelial cells on all sides are not the same. If I'm one epithelial cell, the features here are different, apical features are different. If you are standing with me, lateral features are different, and down where I touch the floor, that is my basal features may be different, because different surfaces have different specialization. So we can say that as far as structure, function, and biochemical characteristics of epithelial cell is concerned, they have different polarity, having different polarities, because there are some other cells in body which are not epithelial cell. All sides of the membrane may have same features. Is that right? Now we'll go into detail of that what are the different features of different domains of epithelial cell and why we need such kind of domains. Let's suppose here is the kidney, right? And in the kidney there are special tubes made of epithelial cells, and these tubes are called nephrons, right? Let me start our example from there. Let's suppose that this is nephron. This is one nephron tube which makes the urine. Right? Now, there are special epithelial cells here. Look at it. These are two epithelial cells. I'm going to make this area of the nephron large. Now, these are two epithelial cells of the nephron. Nephron is a tube, is that right, which is made of epithelial cells. Now, epithelial cells, here what happens? The urine is flowing here, is that right? Fluid filter from the blood and fluid is moving in the lumen. In the end, whatever fluid comes, it is urine, is that right? Now, actually, when fluid is moving in this area, these cells are working. In what way? Let us suppose if glucose is filtered, do you think we should normally lose the glucose and amino acid into urine? No. So from here, glucose and amino acids are reabsorbed and pushed back to the blood. Back to the blood. Am I clear or I'm not clear to you? Right? Daniel? Are you understanding or I should make a better diagram? Okay. Now what we have to see that, let us suppose these are two epithelial cells. This is apical surface, this is basal surface. Is that right? And these are the lateral surfaces. Again, this is another cell with apical surface. Now what the special thing which I want to tell, that let's suppose on apical surface there may be some special feature. For example, there should be some transport system which can take glucose in, or there may be transport system which can take amino acid in, right? So it is taking from the lumen to the cell. Now these proteins or transporters should be kept only on the apical layer. These proteins or special transporters which are present on apical cell membrane should not float into lipids of the cell and come on the basal side. If they float inside and come to this side and they will start, do you think then they will work normally? They will not. Am I clear? So cells on apical side may be working in a different way. On basal side, cell membrane may be working on different way. Now I'm going to explain the specialized feature in different epithelial cells, and I'm going to make one big epithelial cell there, rather two epithelial cells there. Okay, this epithelial cell which I'm going to make here, this is one epithelial cell. And let's suppose this is the top of the cell, the roof of the cell, which is apical side. What is this? Apical side. And this side should be which one then? Basal. And this side, other side, right? And front and the back, that should be lateral domain. And I will put one cell on this side. There were two cells together. I separated them, right? Just to show the specialized feature which we are going to discuss. This is the other cell, the neighboring cell. Now again this cell also has, what is this? Apical side, right? Front, sides, and then it is basal. Now let's call these cells like epithelial houses, right? We can call it epi house. And what are its features? Epithelial houses. If we call them house, this should be the roof, apical side. Now what could be the feature in the roof? First I will talk very briefly. Number one, in some epithelial cell at the roof, there may be special, yes, extension of plasma membrane. Plasma membrane means cell membrane. From cell membrane, there may be special extensions. And these small cytoplasmic extension, right, these may be called microvilli. Microvilli. Then, like microvilli, but there are large cytoplasmic extension, and if feature of the cytoplasmic extensions are similar as microvilli, but these are very large, then we call them stereo. It should be called stereovilli, but unfortunately we call them stereocilia. But actually stereocilia are structurally like villi. right? The thing is that microvilli and stereovilli, which are also called stereocilia, they are structurally similar, except that stereovilli are very long and microvilli are short. Is that right? Then third type of specialization at the top of the cell can be that there is a large extension and there is this cytoplasmic plasma membrane and cytoplasmic extension has specialized feature that this can move, and because this can move, this motile apical structure is called cilia. Remember, microvilli are not motile, stereovilli are not motile, even though they are called stereocilia, but they don't move like cilia. But ciliary structures on the cells are motile. So we can say at the top of the cells there can be chimneys. What are these chimneys? They may be microvilli, not motile, stereovilli, which are not motile, which are also called stereocilia, and true cilia, which are motile. So we will go into detail of every structure thoroughly. Is that right? I will discuss in detail. I will discuss microvilli, then I will go into detail of what is this? Stereovilli, then I will go into detail of cilia. So let's talk about the roof of the cell. Then we'll come to the sides of the cell and then base of the cell. Now, roof of the cell or apical surface of the cell, let me draw the structure of microvilli. Now, what is the basic structure of micro? I'm going into detail of what is this? Microvilli. Now, basic structure of microvillus is, yes. Now, this is cell membrane, plasma membrane. Microvillus is, I've just enlarged this area here. And the classical thing, if you don't remember anything, at least remember that the proteins which are present inside the microvilli, they are the rods of actin. What are they? Yes, these are actin. So core of microvilli is made of which type of protein rods? Yes, actin. This is actin. And of course, these actins, as they go down, right, these structures will go down and they will get fixated, they will get inserted or attached with a network of actin inside the cell membrane, and this network of actin which is present inside the membrane, this actin network is like this. These are actin filaments, right? And these actin filaments, this is actin filament, this is another actin filaments, and there are still some more actin, yes, filaments. This structure, right, under the cell membrane, which is making a network of actin filament, this is called, yes, of course it's part of the cytoskeleton, it is one part of the cytoskeleton, but this structure is called terminal web. What is it? Terminal web. It's a part of the cytoskeleton. Now, this terminal web is held with the plasma membrane with special proteins. And what are these proteins? Spectrins. What are these? Spectrin. So spectrins are the proteins which hold the terminal web with the plasma membrane. And what are these? Actin filaments which are present in the microvilli. They, as they move from the tip of the microvillus towards the cytoplasm, they actually get attached with the terminal web. Inside the terminal web, deeper to the terminal web, there is another group of filaments, and these filaments are called, yes, what are these filaments? I should remind you what? Intermediate filaments. I for intermediate. Intermediate filaments. And then there are some myosin filament also, and these myosin filaments, let me draw it more clearly. This is microvillus here, right? I've analyzed that. Now, there are some myosin filaments, and myosin filaments actually, myosin 1 filament help the actin filaments to attach with the actin filament in the core to attach with the cytoplasmic extension. What is this? Myosin 1 filaments. And then there are special proteins which hold the actin filaments with each other. What are these? These are the proteins which hold the actin filaments with each other, and these proteins are called actin binding filaments. These green are, look, these are actin filaments which are held together with actin binding green filaments, and all this core is held with the outer membrane through myosin 1 proteins, and actin filament as they go into cell, they get attached with the terminal web which is having a lot of actin filaments and few of myosin 2 filaments, myosin 2 proteins, and this terminal web is anchored with the plasma membrane with the spectrin, with the help of spectrin proteins, and there is dangling inside the cytoplasmic deeper area intermediate filament. Any question you have? Now, what is the advantage of structure like this? Actually, this structure simply increases the surface area of the cell. What it will do? You know, like towel, on the towel there are small elevations. The same way the cells, where they want to have more surface membrane, they throw their membrane to multiple extensions. Is that right? So in this way, apical surface of the cell will become, yes, what it will become? It will increase its surface area. Now, this apical surface, which is having a lot of microvilli, this is a feature of the cell of gastrointestinal system. In gastrointestinal system, from the lumen of the GIT, this is for stomach, and I'm just showing one cell here, that when food is coming and going here, to absorb the amino acids and glucose and a lot of substances, we need the cells which have a lot of microvilli. In the same way, when fluid is moving through the nephron, lot of substances need to be reabsorbed. So cells in the nephrons also have a lot of microvilli. These cells or these epithelia which have a lot of microvilli, we say this microvillus is like a brush border, right? We call it brush border, or we call it striated, like striations are there, striated border. So those epithelia which have to reabsorb lot of fluid, right, they throw their apical membrane to lot of folds so that these folds increase the surface area for the absorption, and these folded side of the mucosal cells or epithelial cells, we say these folds are creating a brush border there or striations there. Any question here? Now we come to the stereo. So this is where are the microvilli present? They are present on gastrointestinal cell lining, and microvilli are also present in, yes, rapidly, in nephron cells, especially proximal part of the nephron cell, they have a lot of microvilli, and they are basically actin filaments in the core, and they are not motile. Clear? Now we come to stereocilia. What about stereocilia? This is a good news. You don't need to know a lot about it. Stereocilia are structurally similar with a little difference. Stereocilia are structurally similar with microvilli, right? Stereocilia are very similar with the microvilli, except they are very long. Is that right? And stereocilia, which are very long microvilli, they are also not motile. But these stereocilia are found in which areas? Stereocilia, yes? Who will tell me? Microvilli, I told you, in the epithelial cells which line the gastrointestinal system, and microvilli are also present in the cells which line the nephrons, especially proximal part of nephron. Stereocilia are present in some epithelial cells which are in the inner ear. You remember I told you the specialized epithelium in the inner ear which is concerned with the balance and hearing, right? They have stereocilia. Plus in the males, I mean human males, in the males, in the epididymis, stereocilia are also there. Any question up to this? There's no. Now we come to the third thing. What is this? Cilia, true cilia, which are motile. So let me make a structure of true cilia. Now I will make a true cilia here. This is a structure of microvillus, right? We'll compare it with the structure of a true cilia, right? Let's suppose there is a cell here and it is going to have a true cilia. Actually, ciliated epithelium which is loaded with lot of cilia, that is classically found in the respiratory lining. Epithelium which line the respiratory system, right? That is having lot of cilia, because they, with the cilia, they move the mucus and clear their respiratory airways. Now, what is the structure of cilia? Let me make a big cilium here. Let's suppose we make a mega cilium here. Are they motile or not? Yes. Of course, one cell may have thousands and thousands of cilia, but I'm just showing one cilia, right? Now, what are the special features of cilium? Because it's a motile structure, right? First of all, it should have some strong base. It should have some strong base, and base of the cilium is called basal body. What is it called? Basal body. Let me show you basal body more clearly. Basal body consists of special proteins, and these proteins are, yes, they are as 3 tubes, 1, 2 and 3 tubes. We call it A, B and C. Now this is a triplet. Now this triplet, this is 1 tube, 2nd tube and 3rd tube, right? Now this is one set of triplet, then there is 2nd triplet, right? 3rd triplet, 4th triplet, and total there are 9 triplets. Now there are basically 9 triplets, and these are what? These are special tubes. What are these? These are special type of tubes, and these tubes are called microtubules. Microtubules, which are made of a special protein which is called tubulin. So it's special protein which is tubulin molecule aggregate together, and these tubulin molecules make special tubular structures, and these tubular structures make triplets. Is that right? Now how many triplets are present in basal body? Yes please. 9. This is 1 triplet, 2nd, 3rd, 4th, 5th, 6th, 7, 1, 2, 3. Okay, they should be 9, but actually in my diagram they are showing 7, so I will do a little trick and make them 9, right? Are they 9 now? 1, 2, 3, 4, 5, 6, 7, 8. Oh, now they become extra. Okay. So these are 9 triplets. Now these 9 triplets together, they make the basal body. What they are making? They are making a basal body, and these triplets are held together by special proteins, right? Any question up to this? Now from this basal structure, basically proteins of cilia go out. Actually out of these, how many are these? 3 triplet. Out of this, 2 of them will extend. 2 of them will extend upward. From every triplet, 2 of them will extend upward. So how many doublets will, now one of them is not extending. Out of every triplet, how many tubes are extending upward? 2. So how many doublets will be going up? 9. 9 doublets. Out of these 9 triplets will be going up. So if you make a section here, what you will find, how many doublets are there? 1, okay, let me make it. Now 1, 2, 3, 4, 5, 6, 7, 8, 9. Now from here, how many have extended upward? Yes, 9 have extended upward, but in the basal body they were 9 triplets, but in the true cilium, there are how many? 9 doublets. But inside the 9 doublets, there is central sheath here, and there are 2 singles here. One single is here and other single is here. So we can say in the cilium, the core of cilium is made of tubular extensions from the basal body, and this is made of 9 doublets, right, making the core. In the center of that there is single central sheath which is having 2 single tubular extensions. Any question here? But these are not present here, right? Now these are held, all these doublets are connected with the central sheath through special proteins. These are called spoke proteins. What are these? These proteins are radial spokes, right? Radial spokes which are connecting. This is radial spokes which are connecting these doublets, right, to the central sheath. Then another very, very important feature is that, look, from every tube, right, there are special type of molecules. These are molecules which are like claw, and they are moving outward, and they are trying to catch the next couple. This couple also has its arms going out, and they are trying to catch the next couple. And these special arms, these are special proteins which are extending like arms from a doublet and trying to reach to the next doublet, right? These special arms, okay, let me make it large. I will enlarge this here. Right? This is one tube and this is the other tube. One tube, suppose A tube, and this is B tube. So this is A tube and this is B tube, and they are making a doublet. And from here, this was which protein? Radial spoke protein, which is holding this doublet with the, what is this thing? Central sheath. And from here, there are special type of claws or their arms, and they want to hug the next group. These special arms are called dynein arms. Dynein arms. These dynein arms have special enzyme here which can break down the ATP. So dynein proteins are having capability to break down the ATP. So we call them what? We call them, dynein arms have functionally ATPases, right? And when dynein arm become active, they will hold, dynein arm of one doublet will stick to the next doublet, right? In this way, when the dynein arms act as molecular motors, what they act? They act as molecular motors, and one doublet, multiple dynein arms will hold the next, and that will help the cilium to bend. Is that right? And energy which is derived from the bending, that is derived from the breakdown of ATP by the dynein arms, right? And on the outer side, there are special proteins which are holding these doublets together. These proteins which hold these doublets together, these are called nexins. What are they called? Yes, this is nexins. Let's have a break.